Hello everyone. Before this video gets started, I just wanted to remind everyone that our free online tutoring for the upcoming semester is starting soon. So head over to studystem.org if you need help with chemistry, physics, or biology. Thank you and enjoy the video.
Question number 3 of the AP Biology exam. Researchers hypothesized that the plant compound resveratrol improves mitochondrial function. To test this hypothesis, researchers dissolve resveratrol in dimethyl sulfoxide. The solution readily passes through cell membranes. They add the resveratrol solution to mammalian muscle cells growing in a nutrient-rich solution culture medium that contains glucose. They measure ATP production at several time points after the addition of the resveratrol solution and find an increase in ATP production by the muscle cells. Part A of question 3 says, describe the primary advantage for a mammalian muscle cell in using aerobic respiration over fermentation. So for this, you need to know the difference between aerobic respiration and fermentation. So aerobic respiration happens when the environment of the cell has ample amounts of oxygen, and in that process, more energy is produced. Almost approximately 36 ATPs per glucose molecule are produced in the process of aerobic respiration. But when fermentation takes place, less energy is produced because in fermentation, low amount of oxygen is used for the reaction to take place and almost 2 ATPs are produced per glucose molecule due to fermentation reaction. Now, so the primary advantage for a mammalian muscle cell in using aerobic respiration over fermentation is that in aerobic respiration, more energy is produced compared to fermentation, and you can see that the difference is quite significant. Part B of question 3 says, identify an appropriate negative control for this experiment that would allow the researchers to conclude that ATP is produced in response to resveratrol treatment. Now for this, you need to know what negative control means. So negative controls are particular samples that are used in experiments that are treated just like other samples in the experiment, but no factors affect them. Any change in any factor will not affect the negative control. So the factors will not affect it. And the other term that is used in experiments is called positive control. Positive control samples, they will show some sort of a change due to a change in a certain factor. For example, in this situation, the addition of the resveratrol to the cells is a positive control. And the scientists are observing the ATP production, the change in the ATP production due to the addition of resveratrol, which is considered a positive control. So a negative control for this experiment could be that the scientists must run an experiment without adding resveratrol. So now they would get two samples, one sample with resveratrol and another sample without resveratrol. The one without resveratrol would act as a negative control, and the one with resveratrol would act as a positive control. And by comparing the positive and negative controls, the scientists will be able to determine the change in the ATP production and get suitable and appropriate results for their experiments. The next part says, predict the effect on short-term ATP production when resveratrol-treated mammalian muscle cells are grown in a culture medium that lacks glucose or other sugars. Now, the absence of glucose or other sugars would significantly decrease the production of ATPs. Why would that happen? The reason is because to produce ATP, we use a reaction called respiration. And respiration involves the use of glucose molecules alongside oxygen to produce ATPs. So if glucose molecules are absent, then ATP production will completely stop or decrease. Part D says, The researchers find that resveratrol stimulates the production of the components of the electron transport chain. The researchers claim that treatment with resveratrol will also increase oxygen consumption by the cells if glucose is not limiting. Justify the claim. The electron transport chain is one of the major steps involved in the process of respiration, which is used to produce energy in the form of ATPs. Now, the electron transport chain involves the transfer of electrons. And since resveratrol is supposed to stimulate the electron transport chain, this means that more electrons are going to be transferred. As you might already know, the process of respiration involves the glucose and oxygen molecules and their conversion into energy, carbon dioxide, and water molecules. So if the rate of respiration is increasing by increasing the rate of electron transport chain, then obviously the amount of oxygen you will need and the amount of glucose you will need will also increase. Since the question says that glucose here is not limiting, which means that glucose is in infinite amount, then the oxygen consumption will also need to increase. The oxygen consumption will also need to become infinite to meet the demands of glucose and so that the reaction can properly proceed and get converted into energy, carbon dioxide, and water.